system, the third protoplanet from the Sun became the proto-Earth. It consisted mostly of refractory elements, for most of the volatiles of our solar system became incorporated in the outer gas and ice giant planets. In the proto-Earth, temperatures rose and the planet softened until the inward pull of gravity could even out bulges and dimples, and the body became a sphere. Within the hot proto-Earth, iron started to melt, and because of its high density, this iron sank towards the center, accumulating to form a metallic core. Convecting hot rock remained behind and became the Earth's mantle. This process of separating into a core and mantle is called differentiation. Meanwhile, meteorites continued to rain onto the surface of the proto-Earth until eventually our home planet had cleared its orbit of all other matter. At that point, the Earth had become a true planet. After formation, the Earth cooled and its crust became solid. But continuing meteorite impacts probably pulverized and remelted its crust at various times. One of these impacts, not long after the Earth had formed, was catastrophic. It blasted apart and probably vaporized much of both bodies. Debris from this collision sprayed into orbit around the newborn Earth. Gravity quickly grabbed hold of this debris and pulled it into a new ring. Fairly rapidly, this debris collected into a ball, producing a new body, our Moon. After some time, the Earth cooled to form a solid crust again, but it remained hot enough that volcanic activity occurred, gradually producing the gases that made a dense atmosphere, rich in water and carbon dioxide. Eventually, the Earth became cool enough for most of the water in the atmosphere to condense and collect into oceans. Then, most carbon dioxide could dissolve in the water, leaving a clearer air consisting mostly of nitrogen. Only much later, after the appearance of photosynthetic organisms, did the Earth become rich enough in oxygen to make it habitable for multicellular organisms.
Three-quarters of Earth is covered in water. It's why it's called the blue planet. But imagine Earth without its oceans. And instead, with all the water in the world gathered together in a ball. There's not that much, really. But this water is what makes Earth different from every other planet in the solar system. The oceans have been here for almost 4 billion years, carving the coastline, driving the climate, and controlling the destiny of life itself. But 4.5 billion years ago, when our planet was born, it was just a molten inferno. There was no place for any liquid water at all. Yet the ingredients needed to create water were there, locked away deep inside the Earth when the planet formed. As the first volcanoes erupted, one of the gases that billowed out was steam. And as the planet cooled, it formed clouds. It rained for thousands of years. As the first rivers formed, they began to fill up the lower-lying areas on our young planet, possibly creating the first oceans. But it's reckoned that even this relentless downpour only delivered around half the water that makes up our oceans today. The rest came from outer space. This is a comet, filmed in 2005. It's about 4 miles across, and like all comets, it's made of rock and water in the form of ice. To discover just how much water comets contain, scientists deliberately crashed a space probe straight into it. After it ploughed into the surface, more than 250,000 tons of water burst into space. In Earth's formative years, it was hit by thousands of comets like this. By the time the bombardment finished, it's thought they'd delivered up to half the water in our oceans.
How did life begin? Or at least how did life begin on Earth? This is a question that many of us are curious about, and biologists are working extremely hard to try to figure it out. One possible way this could have happened is that there was a single type of biological molecule that eventually evolved into all of life. Now, a reason to think something like that might have happened is that all life we know on Earth contains the same biological molecules, namely DNA, RNA, and amino acids. RNA is particularly interesting because not only does it encode genetic information, it also can act as a catalyst for chemical reactions. So one hope is that RNA formed in the early times on the Earth, before there was life, and then began to replicate itself. So that sounds maybe very simple, but it's extraordinarily complicated. First, simply by the fact that we don't know what the early Earth looked like. However, there has been some progress in creating subunits of RNA in a laboratory with very simple chemical compounds and ultraviolet light. A subunit of RNA is a phosphate molecule, a sugar molecule, and a base. There are 4 kinds of bases. We can label them with letters, A, G, U, and C. And those letters, in a sense, when you have a strand of them, tell a story. They contain an enormous amount of information, and there is your genetic code. So if in a lab you can see how, just from chemical processes, you can produce a subunit of RNA, and then watch RNA grow, and eventually, if it's possible, have it replicate itself, then you've at least demonstrated the possibility of how life could have formed. Now, of course, life could have started not with RNA, but something even before that, with molecules that don't exist now, say proto-RNA. Or perhaps life actually came from another planet. It landed here on a meteor and it began to grow once it found the favorable conditions of the Earth. We don't know, but our best way of figuring it out is going through the processes of reproducing what may have happened at the beginning of life.
Have you ever wondered how Earth became the only known planet that contains oxygen? Scientists theorize that Earth's atmosphere started off without any oxygen, and that it took billions of years for the right conditions to arise that allowed for the accumulation of air, which supports life as we know it on our planet. 2 studies from researchers at the University of Southern Denmark have been published recently that pertain to the history of oxygen on Earth. By analyzing the chemical makeup of rocks, geochemists were able to find evidence of oxygen levels that existed in the ancient atmosphere. Because oxygen readily bonds with other molecules, there is substantial historical evidence in the geological record of how much oxygen was in the atmosphere at different times. Dr. Donald E. Canfield from the University of Southern Denmark and author of the book, Oxygen, A 4 Billion Year History, said that around 2.3 billion years ago, Earth calmed down enough that the balance tipped in the favor of oxygen. Rocks that are around 3 billion years old reportedly have signs that oxygen was in the atmosphere of our planet at that time, but back then, there was only 0.03% of the amount of oxygen that is found on our planet today.
About 450 million years ago, plants took a giant leap out of the oceans onto the land to become the plants that we know and love today. But how did they survive? Scientists at the John Innes Centre may have found out. Land plants survive by getting water and nutrients from the soil. They do this by forming a special friendship with soil-dwelling fungi called mycorrhiza. These strands of mycorrhiza reach deep into the soil and drag the nutrients and water back to the plants. But when the first algae landed on soil, how did they survive long enough to form these beneficial friendships? New research suggests that they already had the genes necessary for forming this bond whilst they still lived in the sea. Researchers analysed DNA and RNA from some of the earliest known land plants and algae. They found evidence that their shared algal ancestors living in the Earth's waters already possessed the necessary set of genes needed to detect and interact with the beneficial mycorrhiza. The team of scientists believes this capability was pivotal in enabling the alga to survive out of the water and to colonise the Earth. By working with the fungi to find sustenance, the alga showed a clear evolutionary advantage and was selected to thrive in a very different and seemingly infertile environment. This was the watershed moment that kick-started the evolution of life on Earth.
Hey guys, Amy making bad jokes for you on D-News today, but jokes aside, bones are pretty useful, and not just for giving us the internal stability to not be goopy piles of flesh and organs, but because they persist over the ages and give us vital clues as to the history of various species. It's from fossilized dinosaur bones that we've been able to reconstruct what the Earth might have looked like long before humans roamed. And going back further, it's from bones and fossils that we can trace how species have evolved. The fossil record tells us that life started in primitive oceans before the first species poked their heads out of the water and crawled onto land. These are so-called transitional species, and scientists have been able to determine that they first appeared when limbs evolved from fish fins about 360 million years ago, followed by the first land-based tetrapods that showed up around 330 million years ago. But there's a pretty big gap in there scientists needed to fill. To figure out what happened during this 30 million year gap, researchers from the Queensland Museum in Hendra, Australia, analyzed a rare tetrapod fossil from that time period, a nearly 5-foot-long Ocinidus that lived about 333 million years ago. They found from preserved forearm bones that the species could support itself on land. Now, whether it did or did not is a different story, and that evidence came from what might be the oldest known broken bone. In studying this broken bone, the team used computer software to reconstruct the forces required to cause the break. It turns out that it was a lot of force, much larger than the animal itself. And because water acts as a cushion, there's no way such a bad break could have happened underwater. The transitional animal that left that broken bone must have been on land. From reconstructing the break, the team concluded that the animal likely fell from a rock or log about 3 feet off the ground. Of course, they can't be sure, but it's pretty probable given the bone-based evidence. So if the Ocinidus broke a bone on land, it must have spent at least some time out of the water, and that makes it the oldest known tetrapod to be fairly well adapted to life on land, getting us one step closer to nailing down the moment when animals first left the water.
Of all the sea life stalked by Anomalocaris, Pikaia, a tiny worm-like creature, somehow escaped extinction. This bit of an animal had no protective shell or defensive spines. But in the Cambrian period, one part of its design was wholly unique. As Conway Morris discovered, Pikaia has a structure remarkably similar to a modern-day animal called Amphioxus. Both are equipped with a long, stiff rod running the length of the body, the notochord. The notochord, which supported Pikaia's muscle structure, is the design common to all vertebrates. This modest creature may be our earliest recognizable ancestor.
Pikaia, I believe, evolved into something like a fish, and from the fish, of course, we have the amphibians, represented today by the frogs and the salamanders, for instance, which invaded land in the Devonian. And then after that, we have the reptiles and the mammals. So perhaps if Pikaia had not survived, if it had gone extinct at some time, then of course ultimately there would have been no mammals and of course no humans.
Though much about our lineage remains a mystery, the origins of the human backbone are linked to Pikaia and its notochord. Few developments would lead to greater diversity. The reign of Anomalocaris was not short-lived. It flourished for nearly 20 million years, 100 times longer than the entire history of Homo sapiens. But in the evolutionary lottery, it was the lowly Pacaya that survived, bearing offspring that would eventually populate the seas, the land, and the skies. California's Death Valley, one of the most arid regions in the world. 400 million years ago, an enormous river filled this valley, linking it to the distant sea. It was here that the fossils of the first fish to brave the freshwater habitat were found. It had taken them 60 million years to gain access to these estuaries. The man who discovered the fossil was Dr. David Elliott of Northern Arizona University, a specialist on ancient fish.
The rocks have been slid.
Tons of earth and sand formed sediments at this site, deposited by the river that ran through Death Valley eons ago. And it is here that Dr. Elliott has been focusing his excavation and research work on ancient freshwater pioneers.
Can't really tell what that is. This is a pteraspid, this is a lingulid. And lingulids are rather interesting because they're characteristic of estuarine environments, that is, environments in which freshwater and marine waters are mingling. These are areas of very high nutrients where you may have large numbers of organisms coming to feed, and we have many pteraspids here. They're also areas that many marine organisms can't get into. They're not able to survive in brackish conditions, and so it may be that pteraspids were protected from some of their possible predators by living in these environments.
Freshwater habitats have almost no salt content. A change in salinity can spell instant death for unacclimated marine life. This paramecium thrives in the salty medium of seawater. Breathing saline fluid, it regulates its mineral levels and ejects wastes. Salt-free water is added, changing the balance. In this unfamiliar, hostile freshwater environment, the paramecium takes in water without voiding it, destroying the cell. The now bloated paramecium ruptures and dies. Pteraspis is about 8 inches long, its head covered with tough bony armor and its body with primitive scales. Unlike paramecium, Pteraspis managed to adjust to this new freshwater habitat with a little creative bioengineering. The pteraspid's protective shell prevented water from penetrating the body through its outer layers, but the gills it used to breathe were a weak spot. To balance its system, Pteraspis developed a kidney powerful enough to pump extraneous water from its bloodstream. Transforming its physiology was a major evolutionary breakthrough for the first freshwater fish, but it was only a first step. "400 million years ago, life on the river was unfolding. But the freshwater habitat posed new challenges, rapid currents and waterfalls to negotiate. It took millions of years for marine life to adapt to these new ecosystems. To survive here, Earth's first fish would experiment with many anatomical designs. Miguasha Park lies on northeastern Canada's Gaspé Peninsula. 400 million years ago, this was a tropical region, situated directly under the equator. Etched in this meandering stretch of cliffs is the 20 million year history of fish evolution. Fossils of freshwater pioneers, fish with fins to resist river current, carnivorous fish with jaws and teeth,
evenly distributed. There's some layers with more species.
and a peculiar fish entirely different from any of the others. This fish had a backbone. Until now, there were fish with tough exoskeletons, but none with hard bone inside the body. Cheirolepis, the earliest creature with a spine, appeared 390 million years ago. The most common fish today, the bony fishes, are its direct descendants. Like modern fish, Cheirolepis was equipped with one pair each of pelvic and pectoral fins. It was sleek, with jaws and sharp teeth for catching prey. Acquiring a spine, Cheirolepis developed strong muscles to swim with speed and power. But there were other agile swimmers in the river that had managed to do without one. Why then did the backbone develop? While the sea was rich in minerals essential to life, the river was in short supply. This is heart tissue. Working in unison, these cells allow the heart to pump blood throughout the body. To function properly, the heart needs calcium. Calcium concentration in a river is in perpetual flux. When levels are high, calcium is stored as bone. When they're low, the bone reservoir supplies the necessary mineral, regardless of external conditions. Meanwhile, the seemingly solid bone is constantly being replenished. At the top of the screen, hard bone is visible. Moving below are cells that dissolve it. Once the bone is dissolved, calcium circulates through the bloodstream to every part of the body. The fate of ancient fish rests on a simple backbone. Those without eventually perished from calcium deficiency or returned to their native sea. For many years, Cheirolepis reigned as the king of freshwater fish. His anatomy gained him independence from the marine world. Today, the descendants of Cheirolepis thrive in both fresh and saltwater habitats. The river was a proving ground for the earliest fish and their offspring. And in this new habitat, they gradually adapted the body system's key to survival on land. Another fish with a backbone appeared on the river at about the time of Chaerolepis. It was called Eusthenopteron, a bottom dweller. Eusthenopteron was not an efficient swimmer like many fish, but it had secured an evolutionary niche with powerful bony fins to push its way through the river's undergrowth. This fish was a stealthy hunter poised for action. Eusthenopteron's habitat shaped its unusual bone structure, which would one day lead to the evolution of terrestrial animals. South America's Amazon breeds acres of jungle swampland, mimicking conditions in the age of Eusthenopteron. And with the swamps, a host of unusual life forms. Among them, the lungfish, a mud-dwelling creature. This fish has gills, but it also has lungs for breathing air. In such murky water, oxygen content is often very low. The lungfish probably developed lungs to breathe oxygen from the air when the supply in the water was depleted. Just as his ancestor Eusthenopteron had done eons before. Poking its head above the surface for a life-sustaining breath, Eusthenopteron surely caught a glimpse of the world above the waterline. Now that fish had evolved kidneys, bone, and lungs, land was